Hey guys, all right, we're back. Let me pause the Olympics. All right. Okay, sorry. All right, so we're back. We are ready to rock and roll. Let's do this last chapter before the exam on Wednesday. 50 questions, 20 extra credit per your request. So hopefully this will get you guys rocking and rolling and feeling better for your total exam grades. All right, let's do this. So we got the flow of genetic information. Remember we talked about from DNA, which obviously is in the nucleus, to messenger RNA, which is a single strand right outside of the cytoplasm. And then, of course, this is going to convert it to a sequence of amino acids or a protein connected by polypeptide bonds. In this case, very super strong bonds compared to that DNA with weak hydrogen bonds. So that protein is going to be super hard to actually unravel. All right, so we think about albumin, think about keratin, think about things that end in IN. Those are usually proteins, very, very important, especially for structural portions, everything in the actual cell itself. Okay, so we got to know what transcription is. Transcription is when you synthesize a single strand of messenger RNA under the direction of DNA. Okay. Translation is when you take that message and you translate it to a single protein, right? The proteins themselves have primary, secondary, tertiary, and quaternary structures. Do you guys remember that? Primary structures were just the sequence of amino acids. Secondary structures were those alpha helices and beta pleated sheets. Tertiary structures was a globular protein. Quaternary structure were two alpha and two beta pleated sheet. So proteins don't just come straight out of the oven like a immediate cake. No, it needs to go bake for a bit. You need layers of that, 4 layers of that before it's going to be a functional protein. So translation is a totally different process because you're translating that message into that sequence of amino acids or a protein. Transcription we talked about in class today, which is DNA, which is transcribed in the S phase of interphase or synthesis, essentially to allow for a single strand of messenger RNA so that can fly out of the nuclear pores, okay? All right, so currently a gene is just a region of DNA. It's going to be expressed to produce a functional product that is either a sequence of amino acids connected by polypeptides or a single strand of messenger RNA, transfer RNA, a ribosomal RNA. So what's transcription and what's translation? Transcription is what? From DNA to messenger RNA. Translation is to take that message into a sequence of amino acids, also known as a protein. So the overall story is, here is the nucleus of the cell. The DNA, which is double helix, is going to be what? Transcribed into a single strand of messenger RNA. That's good enough to fly out of those nuclear pores so the ribosome can attach to it. Once the ribosome, which has a top and a bottom subunit attached to that message, the message is going to be translated into a sequence of amino acids, also known as a polypeptide. So this guy becomes primary. This is just a primary sequence right here. So you can see it's like a train or caboose of single strand messenger RNA. These guys are individually, sorry, single strand of polypeptides. These guys are essentially going to turn into folds like alpha helixes and beta pleated sheets, then the tertiary, then the quaternary protein. Okay, this is just a bread mold. It's showing you here that it's growing in culture. The reason why we're showing this one is to show you that the amino acid sequence of even penicillin or bread mold, which is a type of fungus, even that guy contains a written sequence of codes that directly is going to result into a sequence of amino acids for your protein. All right, so when you construct a protein, what do? You basically translate it in a certain language to that sequence. So this is what you have to do. There's a triplet code in that message. The message usually is like AUG, which is the start codon. AUG translates into methionine, which is a start amino acid for the 20 amino acids in the body, right? And it essentially is a codon or a triplet base pair like AUG. The other ones could be GLY or glycine, or it could be aspartic acid or ASP. Whatever they are essentially three key words called codons or a triplet base pair. So it says, what's the minimum number of nucleotides necessary for a codon of 100 amino acids? So if one amino acid, which is like methionine, codes for AUG, that's 3, right? 3 times 100 is 300. So that's how many nucleotides are going to be in that sequence of amino acids. This is just showing you here how we exactly get to that amino acids. The DNA molecule is transcribed, right? It's in this case carrying sequences of genes. Those genes themselves are opening up into your DNA strand. Once this guy is transcribed in the nucleus, it's going to fly out of the messenger RNA, as a single strand. This single strand, you guys can see UUU. That's going to create a specific code, this triplet base pair, for one certain amino acid. And this is like a square shape just to show you that this is specific for this code. GGC is the next triplet base pair. This is going to translate into this shaped amino acid. So you can tell the next one, CGU, is translating into this amino acid. Either each amino acid is going to be attached by these super strong bonds called polypeptides. So this is going to create your sequence of amino acids or your primary sequence that's going to then turn into your secondary sequence, which is again the alpha helixes and beta pleated sheets. All right, this is kind of cool. This is like the cat on the left, which is engineered with a protein to fluoresce red, whereas exposed to this guy on the right-hand side, he doesn't have a protein engineered that fluoresces when it's exposed to UV light. So we can actually manipulate these poor little cats to, in this case, fluoresce in the presence of a UV light. Okay, so transcription is what? You're taking the DNA, you're transcribing it in the nucleus, and you're creating a single strand of messenger RNA. Do you guys remember DNA polymerase? Now we got RNA polymerase. RNA polymerase is going to line up those nucleotides. They're going to join along a single strand of DNA, and they're going to follow the normal base pairing rule we have, which is A to U, G to C. Notice I said U. U is used in translation, and in this case, that's going to be the code for that is specific strand. So you got a specific nucleotide sequence. It's going to start at the promoter region. The promoter is like the start, kind of like the one that starts the whole thing, right? He promotes it. He's going to be the binding site for RNA polymerase, kind of like DNA primase was the binding site for DNA polymerase, and he's going to determine where transcription starts. So RNA polymerase will add RNA nucleotides until it reaches the terminator. When it terminates, the terminator, it's going to single the end of the gene sequence, and that's it. So you got the promoter all the way to the terminator, and this is going to be, all right, I'm done with actually copying it into single strand of messenger RNA. So the question is, how does RNA polymerase recognize the start and the end? Again, because of the promoter, which is the start, and the terminator, which is the end of the gene sequencing. So let's take a look at the whole picture. This is a couple of things. We got to initiate it, and then we're going to elongate it. Oh, God. Okay, where'd we go? So one of the first things you got to do is you initiate the sequence. So RNA polymerase will attach to the DNA at the promoter region. It will go down the length of it, and it'll essentially base pair up until it gets to the terminator. In the end, you're still going to get a single strand of messenger RNA. This guy is going to create a replication bubble. Anything unused is going to be cut out. This guy is going to be the newly formed RNA strand. You can see it's purple. That's going to basically be the codon to carry the code from that template DNA strand. Because you can't pull out that DNA. It's double helix. If you pull it out, oh, that guy just totally flopped. Sorry, I was watching the Olympics. Oh my God, he totally flopped. He's so disappointed in himself. That's okay, bro. You're only at the Olympics. All right, so the template strand itself is that original strand. The RNA is the codon. So it will carry the code from the DNA and then fly out of the nuclear pores. So when you initiate it, then you got to elongate the strand. In the end, oh God, why does it keep doing that? In the end, what you're going to see is what you're going to see is the elongation of it with the help of RNA polymerase, which is going to open up that bubble and allow for the newly made RNA single strand to form as a result. If you take a look at the direction of transcription, it's still 5' to U'. And there's that uracil I was talking about. If this is the original strand, TCA, we know it's GAU, right? So if this guy is going to base pair up, here it is, ATA to GAU. Notice uracil will pair with adenine, and if you keep going down, you see uracil pairing with adenine. There's those free nucleotide pairs. So you're basically going to pull out, especially in translation, you're going to, why does it keep doing that? You're going to essentially create a single strand of complete RNA. The terminator is when it gets all the way towards the end of the sequence and the RNA polymerase detaches and you get that completed mRNA transcript, okay? You are not done because we got the video.
The first step in protein synthesis is transcription of RNA from DNA. The portion of the DNA that is The first step in protein synthesis is transcription of RNA from DNA. The portion of the DNA that is transcribed into an RNA molecule is called a transcription unit. An enzyme called RNA polymerase carries out transcription. With the help of protein transcription factors, it attaches to the beginning of a region of the DNA called the promoter, pries the DNA strands apart, and untwists a short portion of the double helix. RNA polymerase moves along the DNA, pairing up RNA nucleotides with their DNA complements, adding nucleotides to the end of the growing RNA molecule. Here's a close-up view of elongation. Only one DNA strand, the template strand, serves as a template for RNA synthesis. RNA polymerase moves along the DNA strand in the 3' to 5' direction, adding nucleotides to the 3' end of the RNA chain. Note that U in RNA pairs with A in DNA.
All right, awesome.
Once transcription is complete, RNA polymerase releases the completed RNA and detaches from the DNA.
There it is, that messenger RNA prescript. It's ready for the introns to be excised, the exons to be kept. Okay, talk about those in just a second as well. All right, so when you're eventually transcribing it from a DNA double helix in the nucleus to a single strand of messenger RNA, that messenger RNA carries the message, right? That genetic message from DNA has got to be perfect. So that single strand is really what's used to be translating the message into that sequence of amino acids, right? Okay, so when the picture of that pink mRNA prescript came out, what happened was there's too much genetic sequence in it. So sometimes what we have to do is cut out the stuff that we don't need. Those are called the non-coding segments. So if you splice out or cut out with a spliceosome the introns, then you're going to keep the exons. So let me repeat that. The pre-mRNA strand that came out, that little pink strand that we eventually transcribed from the original DNA strand, what you're going to do is there's sequences of it called introns and exons. The introns are excised. I know it sounds like backwards, but the introns are excised and cut out with a spliceosome, and then all the exons, the leftover part of the pre-mRNA transcript, is kind of added together, and then you have a poly A tail at the end of it. So you cap the beginning, put a tail at the end, now you got to have a fully complete mRNA transcript. Let's look at the picture of it. Very, very important. This is the pre-mRNA transcript. Here's all the exons. We're going to keep those. The introns are cut out, right? The introns are cut out. So then you got to glue the exons back together. So before you do that, first you got to cap it, and then you add a tail to it. Notice the introns fall out. They're spliced out. What you add together only is just the exons. Once the exons come together, you put a cap, a poly A tail, and then this is the guy that's going to fly out of the cytoplasm. So that's why I called this guy the pre-mRNA transcript, because he's just the beginning. You still got to excise the introns, add the exons together, cap it, put a poly A tail, then that dude's going to fly out of the cytoplasm. Okay, so when we do this, we're going to have to translate the message. So now the mRNA transcript has come out. He's happy, he's got the message, the introns are out, it's capped with a poly A tail, he's flying out of the nuclear pores. The mRNA is this is perfect. It's got the message, it's nice and short, right? It's not super long, it's not that double helix of DNA, he's not kept with introns, he is cut, he's spliced, he's capped, and he's tailed. So now we have that pre-mRNA transcript, which is then finalized into mRNA. This guy is the code. We call him the codon. The code needs a anticodon. So he needs to pair up, right? If you got to translate that message, the code itself has to be paired with an anticodon. If you add the anticodon to it with the codon, this guy is going to give you that sequence of amino acids to give you that protein, right? Again, it's not that easy. We're just going to, boom, make a protein. That codon is the mRNA transcript that had what? The introns excised, the exons kept, capped and poly A tailed. Now you have to have a tRNA or transfer RNA come up. He looks like a clover leaf. He's going to fly up and bring an anticodon to pair with the codon. Okay, so what's going to happen is there's your clover leaf. This is your transfer RNA. Kind of looks like a cool clover, doesn't it? He's going to have an attachment site for the amino acid. Like ACC is going to code for a specific amino acid. So this dude's flying around, right? The transfer RNA molecule has that simple attachment site to show you, hey, I can be specific for every amino acid out there. And this is the end where either methionine, aspartic acid, leucine, glutamic acid, whatever it is, will appear. Now, each of those, leucine, aspartic acid, glutamic acid, whatever they are, they will have a specific triplet base pair, okay? It's going to be the anticodon that's going to pair with what? The codon. What's the codon? The mRNA transcript. So let's see how this works. There's bonds between this tRNA molecule. There's the sites. Here's what it kind of looks like, the transfer RNA molecule. It's going to have the enzyme attached with the amino acid and transfer RNA. That's the 3D version. And it's going to have rRNA. Ribosomal RNA does what? He's going to be the helper molecule to make the proteins. So now we've got what? We've got the mRNA molecule, which carried the message. You got the transfer RNA molecule that transfers what? It carries the anticodon, which is what? The amino acid with the protein attached to it. Then we have ribosomal RNA, that the protein is going to come together into primary, secondary, tertiary, and quaternary sequences. Let's see if we got this whole picture together. Awesome. Here's what it actually looks like underneath a 3D imaging. This is your ribosome right here. You open it up, it looks like a Pac-Man. It's got sites. It's got a P site and an A site. The A site's for assembly. The P site is for final assembly, and then it's going to spit out, churn out, pop through his Pac-Man mouth. He's going to make this guy, which is a sequence of he's going to make this guy, which is a sequence of amino acids, which is your growing polypeptide, which is going to turn into your protein. So once the mouth opens, your mRNA transcript, which had what? The original code, remember? So the original code that came out of the nucleus is going to bound with that ribosome. The ribosome is going to clamp onto any part of it, and each of it is going to contain an area for the transfer RNA with a growing polypeptide to come up. The transfer RNA is highly specific. It's not going to code for anything else except methionine. This transfer RNA is not going to code for anything else except leucine. Another transfer RNA is not going to code for anything else except glutamic acid. So transfer RNAs are floating around everywhere with that specific amino acid attached to it. He'll come and only bind, because it's an anticodon, to the codon. Once he binds, he's going to fly out, but not unless it's going to continue to grow a polypeptide or a chain of events. So I can't wait for you guys to see the video. This is the upper larger subunit. This is the lower subunit. This guy is your transfer RNA molecules with your messenger RNA codon. It's going to spit out, in this case, your growing polypeptide. The anticodons are sitting right here. Once it cycles through, he's going to basically spit out your polypeptide. These are your sites for the ribosome, the site of protein synthesis. You have the A site, it starts at first. You got the P site, and then it's got like an exit site, right? That pops out, in this case, the growing polypeptide. He's got the large upper subunit, the smaller subunit. He's going to open his mouth like a Pac-Man and grab onto what? The mRNA transcript, which was what? It was that sequence, that original code that came with everything. So now we're starting to see the original codon, which is your mRNA transcript. This is the transfer RNAs that are just basically going to attach. Once this is open, it's going to be specific for a certain amino acid. And then when it keeps growing, it's going to basically base pair up at every single triplet base pair for the mRNA codon to the anticodon carried by the transfer RNA. These little green things are the anticodons. It's going to be highly specific for one of these growing polypeptides. Okay, so 3 things we have to remember by translation. You have initiation, elongation, and termination. You got to initiate your original what? When you take your poly A tail at the end and you got the cap, this is your mRNA transcript, right? You got the start of the genetic message, 1, 2, 3. This is the codon, right? This is the one where we excised the introns. We kept the exons and added all the exons together. We capped it. We put a tail on it. This is what? Your original codon that flew out of the nuclear pores. Very important because that is the codon where the transfer RNA carries the anticodon. He will also have the methionine, aspartic acid, glutamic acid attached to it, certain transfer RNA. He's going to bear the amino acid necessary for you to elongate this guy. So what would happen if a genetic mutation occurs in a gene, change that start codon to some other codon? Well, if that's going to happen, the whole thing's non-functional and initiation is not going to start. Okay, so here we go. We got the start codon. The mouth is open. Here is the mRNA transcript. The transfer RNA, remember I told you MET is methionine. He is the start codon. Everything in the body starts with MET. MET is the initial amino acid or methionine. Don't forget methionine is called the start amino acid or the start anticodon that is picked up. He is UAC. He's going to base pair up with AUG. AUG codes for what? Methionine. So if you keep going on, you notice AUG with UAC, A to U, U to A, C to G, no more thymine at all. Has both the P site, he's going to move over to the P site. Then MET is just going to keep growing. The next one, aspartic acid is going to attach, glycine, leucine, any specific amino acid. It's going to be totally different because the sequences are very, very unique. So as elongation happens from the promoter sequence to the codon where the stop codon is, you got a stop codon that is, instead of AUG, it could be UUA or UAA. Because it's UUA or UAA, that guy is called the stop codon. So you got a start codon with methionine and you got a stop codon with UUA and UAA, okay? So here it is. You got to recognize the code, the original mRNA transcript, which is the code from the nucleus, right? It came out of the nuclear pores. It's flying out in the cytoplasm. You have the ribosome notched onto it, and the ribosome's going to do what? He's going to introduce these A and P sites where the transfer RNA is going to find a spot to actually attach, right? And then once it attaches to the P and the A site, you're starting to see a growing polypeptide with these peptide bonds forming. As they form, it's going to translocate. Your tRNA is going to move, fly around. Here's your little purple little area for another amino acid to be picked up. He's just going to keep flying around until he needs something else to attach to. He's going to keep flying around until he needs something else to attach to. So this is going to be the next mRNA movement. In the end, you're going to see a growing polypeptide, which is a sequence of amino acids connected by super strong polypeptide bonds.
In translation, a cell reads an mRNA message and assembles a polypeptide accordingly. We can divide translation, the process of building a polypeptide, into 3 stages: initiation, elongation, and termination. Translation is initiated when the small ribosomal subunit binds to the leader at the 5' end of the mRNA molecule. The anticodon of the initiator tRNA binds to the start codon, AUG. The initiator tRNA always bears the amino acid methionine. Proteins called initiation factors help bring the mRNA, the initiator tRNA, and the small ribosomal subunit together with the large ribosomal subunit to form an initiation complex. The initiator tRNA sits on the P site on the ribosome.
That was awesome. Oh, good.
Elongation of the polypeptide now occurs, with amino acids being added one by one. The codon in the A site of the ribosome pairs with the anticodon of the appropriate tRNA molecule.
See valine?
Part of the ribosome
Peptide
catalyzes the formation of a peptide bond between the amino acid extending from the P site and the amino acid extending from the A site. The tRNA in the A site now translocates to the P site. The tRNA that was in the P site moves to the E site and exits from the ribosome. Meanwhile, a new tRNA brings its amino acid to the A site and the process is repeated.
Perfect. That's tyrosine attached to arginine, valine, and methionine, all amino acids. So transfer in RNA does what? He drops off the anticodon.
3 special base triplets, UAA, UAG, and UGA, do not code for amino acids, but instead act as stop codons, which terminate the process of translation. A protein called a release factor binds to the stop codon in the A site. It acts to free the completed polypeptide from the tRNA that is in the P site, and the translation assembly comes apart. The polypeptide becomes folded, modified, or combined with other polypeptides to form a functional protein.
Okay, that was an awesome video. So keep in mind, methionine, valine, arginine, tyrosine, serine, and threonine, these are what? Amino acids connected by strong peptide bonds. This is your primary sequence of amino acid. What was this down here? That was your mRNA prescript, right? Your transcript that had what? It had introns excised, and all of these are the exons. What was it eventually turned into? A cap and a poly A tail. This is the one that flew out of the nuclear pores, and then a ribosome, the large subunit, small subunit attached to it, with the E, A, and P sites. This is going to be the location where transfer RNA, remember those guys that you saw just fly around? Those transfer RNAs keep flying around in the nucleus, in the cytoplasm, right? All they do is they carry these amino acids. So when they come and find its codon, this is the codon here, they're bringing the anticodon. That anticodon pairs with the codon, and it's going to attach with an amino acid, and it's going to keep growing and growing until you get to what? The stop codon. The stop codon does what? Release the entire sequence of amino acids back in this case to your final protein. Okay, good job. So DNA is transcribed under the process of transcription into messenger RNA. The message is translated into a protein. So the sequence of codons in DNA via the sequence of codons in mRNA spells out the primary structure of a polypeptide. Okay, that's a sequence of amino acids known as a polypeptide. Which of the types of nucleic acids you've learned about does not participate directly in translation? Remember, we had all the RNAs, right? Messenger RNA, transfer RNA, ribosomal RNA. So what's the only nucleic acid left? Those are 3 RNA molecules we talked about. The only one left is DNA. So that's the only one that doesn't participate in translation. What does DNA participate in? Transcription. All right, good. So here we have everything together again. This is transcription plus translation. Let's blow this guy up just a bit to pull it out. Transcription occurs where we're going to take that DNA and create a single strand of messenger RNA with all the introns and exons. The introns are the gross stuff. We're going to excise them later, but this guy is the first stage of transcription. He's going to eventually end up in the cytoplasm, where you're starting to see transfer RNA carried by an amino acid. These anticodons are going to pair with what? The codon from the RNA, the messenger RNA. So he's going to come and find that site where the ribosome says, this is the site for protein synthesis, come join me. So there's going to be the start codon of AUG, and the transfer RNA is flying in the anticodon of UAC. UAC is going to code for, it doesn't even tell you here, this kind of hexagon-shaped polypeptide, right? So this guy is going to fly out. He's also an amino acid. He's going to keep growing until he exits. So the transfer RNA eventually disappears, and the next one comes and finds the next series of codons or triplet base pair. The anticodon comes in to the ANP site. He is basically going to attach on and then form a growing polypeptide. In the end, the stop codon, if it's UUA or UAU, whatever it is, will stop this actual translation of the messenger RNA, which is that original codon. So then you're going to see a free polypeptide float all over the nucleus, where it's going to basically be used by the body internally, or you can excise it out to a new cell. So the story is there could be lots of mutations that happen with transcription and translation. Or there could be chemical or physical agents called mutagens that can be caused by either replication problems or recombination issues. So if these mutagens happen, sometimes what happens is you can substitute a nucleic acid substitute a nucleotide like an adenine or thymine, guanine, or uracil, or you could insert it or delete it altogether. If it deletes, inserts, or substitutes, that nucleotide substitution can result in a shortened product. And what happens is it would prematurely terminate the polypeptide, and then you're in trouble, right? So this is normal, let's say, blood. This is mutant blood, also called sickle cell anemia. So in normal blood, you got this protein called hemoglobin. Hemoglobin is what binds all your iron, and it attaches your oxygens. So you basically get this quaternary oxygen attached to hemoglobin and protein. So that's going to become a functioning red blood cell. So your erythrocytes or red blood cells has hemoglobin or protein. If this is the normal DNA molecule of CTT, okay, the mRNA transcript is going to what? He's going to transcribe it into GAA. So that's normal. What happens with a mutant is instead of CTT, there's one substitution of that thymine with adenine. So the mRNA transcript becomes GUA. That single nucleotide base pair then turns into GUA, which is the codon that codes for the anticodon of tRNA, which carries the amino acid of valine. So instead of glutamic acid, it has valine, which is then going to cause sickle cell anemia. So sickle cell anemia is that sickle shell-shaped a red blood cell. He looks like you guys have seen the Grim Reaper, right? Holding its little sickle cell, like long like stick, right? This guy is that kind of shear that cuts, right? So this guy is so small that he's not going to hold that many oxygens. So folks with sickle cell anemia actually become very lethargic because they can't hold 4 oxygens like normal hemoglobin does. Sickle cell hemoglobin is only going to carry 1 or 2, which is why those folks with sickle cell anemia become very tired, lethargic. A normal 9 to 5 workday is going to be exhausting for them. So with a normal gene, let's say with mutations, if you substitute, let's say here, you substitute this guanine with this adenine, it's going to code for serine. If you delete something, this delete altogether a uracil, it's going to move over that G right up here, and then it's going to code for leucine. If you insert something brand new, now you're going to create what? A super long series of brand new amino acids, which is going to be the worst thing possible. So here is substitution. You're substituting just that G, guanine, for the A, which is going to change that one sequence of amino acids. This guy is if you delete and throw away one of these uracils, so then everything moves over, like this guy just moves over. So the G is there instead of, oh God. So the G is there instead of, what the heck just happened? So the G is there instead of, oh, that's insertion. The G is there instead of the U, and then that throws off the entire sequence, and then you get leucine and alanine. And then when you insert something brand new, everything shifts. So leucine, tryptic acid, and arginine end up becoming the new sequence of amino acids. Okay, viruses and bacteria. Let's take a look at viruses and bacteria. Virus, remember, contained what? The protein outside and the DNA inside the head, right? So a virus is just an infectious particle. It consists of a little more than genes in a box, a bit of nucleic acid wrapped in a protein coat called a capsid, and in some cases a membrane envelope. When a phage DNA enters a lytic, remember lytic means lyse, cut, cycle inside a bacterium, it is replicated, transcribed, and translated. Remember I told you guys the DNA of a virus takes over the DNA of the host and it cuts it and makes its own DNA. So the new viral DNA and protein molecules then assemble into new phages, which lyse through the lytic cycle. They burst out of the new cell, killing the cell, and then turning to new cells, killing the host cell and turning to new cells. The lysogenic cycle is when the DNA inserts into the host and it's passed on from generation to daughter cells. So once it's inserted, the DNA is referred to as a prophage and most of its genes are inactive. Later, it may initiate phage production. So describe one way a virus can perpetuate its genes without destroying its host. Well, what type of replication is that called? Well, some viruses can insert their DNA into chromosomes, which replicate the viral gene, and when it replicates its viral gene, this is called the lysogenic cycle. So here we're going to talk about both cycles, the lytic and the lysogenic. The lytic, which is when it cuts it like this, and then the lysogenic when it just creates many, many, many cells. So the virus has two phases, the lytic and the lysogenic cycle. If you have that phage, that bacterial virus attaching, once it attaches, it's going to inject and create its own circular piece of DNA and not create its normal bacterial DNA. That phage is in control. Once it does it, the circular piece of DNA undergoes either two things. It can either go the lytic cycle or it can undergo the lysogenic cycle. So in the lytic cycle, you get new phage DNA and new proteins are assembled, and you got this lysis where it breaks open that bacterial DNA and releases all those baby varioids. If it goes through the lysogenic cycle, what happens is the phage inserts itself. Remember I told you here, it just takes it over and kills everything? Here, the viral DNA can insert itself into the bacterial chromosome. So there's that light blue into the old original circular DNA. And this guy will basically replicate, split, 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 copy, copy, copy the bacterial cell. And then you're going to get this breaking many cell divisions of viral DNA over and over. So this is why viruses are pretty scary. If they can actually get inside that DNA, it will either go through a lytic breakage cycle where it essentially kills it and releases new phages, where then those hundreds or 200 phages can attach to new hosts, or it can take over the circular DNA, attach a prophage, and basically copy, copy, copy many, many cell divisions. So there's a lytic cycle. Again, it's going to cut, break, degrade, releasing hundreds of varioids, or it can go through the lysogenic cycle, which means that it's going to insert itself into the bacterial chromosome. It's going to undergo replication many, many times over, and now you start to see a major, major problem.
In contrast to the lytic cycle, In contrast to the lytic cycle, the lysogenic cycle reproduces the viral genetic material without destroying the host. The lysogenic cycle of phage lambda begins when a phage binds to the surface of a host bacterium. The phage injects its DNA into the host cell, leaving the empty protein coat outside. The viral DNA is incorporated into the host cell DNA, where it is called a prophage. Every time the host bacterium reproduces, it replicates the phage DNA along with its own and passes the copies on to daughter cells. Occasionally, the phage DNA exits the bacterial chromosome and initiates a lytic cycle. The viral DNA takes over the metabolic machinery of the host cell to make phage DNA and proteins. The host cell lyses, releasing phages which can infect other cells. This diagram summarizes the lysogenic and lytic cycles of phage lambda.
All right, so that was pretty scary, right? Here in this case, if it injects its viral DNA, it will take over the overall DNA of the cell. Sometimes it will just split into the lysogenic, sometimes it'll go back into lytic. So this is like a double whammy of lytic and lysogenic cycles. And in this case, if it keeps replicating, or in this case it lyses, sometimes the phage can get out of that circular DNA and then boom, just start to break open that cell itself, that host cell, which is why it's pretty scary that viral DNA have such intelligence to do all of that. Okay, we're almost done. Don't worry, we are almost done. So this is, again, that phage of T2. Let's see what this does.
Bacteriophages or phages. Bacteriophages or phages are viruses that infect bacteria. This is a T4 phage, which consists of DNA inside a protein coat. The lytic cycle begins when the tail fibers of the phage stick to receptor sites on the surface of a host bacterium, such as E. coli. The phage injects its DNA into the host cell, leaving the empty protein coat outside. The DNA of the host cell is destroyed, and host cell enzymes and nucleotides are commandeered to replicate the phage DNA, making more phage DNA. The host cell's enzymes and ribosomes transcribe the phage genes and translate them into phage proteins. Phage parts accumulate and assemble to form phages. A phage enzyme digests the bacterial cell wall and the cell ruptures or lyses. As many as 200 phages spill out. Each of them may go on to infect another cell. This diagram summarizes the lytic cycle of bacteriophage T4.
Okay, so T4 does what? Attaches, takes over, kills the original DNA inside that host E. coli, and it creates more of its individual virions, which turns into a lysis, and then it's going to continue to infect more and more cells, right? And we talked about the lytic cycle as a primary way that will take over that cell. All right. We know there's lots of viruses, right? Many viruses can cause disease in animals and in plants. So viruses can attack plants and obviously kill the neighboring plant as well. So flu viruses and plant viruses have RNA rather than DNA as their genetic material. Some animal viruses steal a bit of the host cell membrane as a protective envelope. So it's even scarier. Explain how some viruses replicate without having DNA. And some, again, many of these viruses have RNA, which replicate inside the host cell by special enzymes encoded. The viral DNA serves as mRNA for the synthesis of viral proteins. So here we have viral DNA. This is a RNA virus. So here we have the actual viral RNA genome. This is going to allow for entry directly by taking, doesn't it look like the phospholipid bilayer? It's going to actually have these spikes on the outside, and it's going to have a membranous envelope that when it opens up, it's just going to join into the actual cytoplasm itself. It's going to uncoat and create that viral RNA. It's going to synthesize into an enzyme, and then you're going to start to see normal RNA synthesis from the template to the normal viral DNA or normal viral proteins, which are going to reassemble again and then exit out. So here we have the virus showing you the...
A virus is an intracellular parasite that can reproduce only by taking over a host cell. A virus consists of a nucleic acid genome enclosed in a protein shell called a capsid. In the virus shown here, the genome consists of DNA, but some viruses have RNA. Some viruses are also covered by a membranous envelope that is derived from the membrane of the host cell. There is usually a lock and key fit between the proteins of the capsid and receptors on a particular type of host cell. The virus attaches to a host cell and viral DNA enters the cell. The viral DNA uses nucleotides and enzymes of the host cell to replicate itself. The viral DNA then commandeers other host cell materials and machinery to transcribe its genes into messenger RNA and translate the RNA message into capsid proteins. Viral DNA and capsid proteins then assemble into new viruses. Mature viruses leave the host cell, often destroying the cell in the process. The viruses can go on to infect other cells, spreading the viral infection.
Wow, okay. These videos are awesome. So definitely just watch all the videos, especially when you're studying for the exam. Just keep watching them. So we got HIV, which is the human immunodeficiency virus, the virus that causes AIDS or acquired immunodeficiency syndrome. Please know that there's other emerging viruses that seem to burst into the scene out of nowhere. So all of a sudden the medical community has to deal with this. So the question is, why doesn't a flu shot 1 year give us immunity to the flu in subsequent years? Come on, you guys know this. You know the viruses evolve rapidly by frequent mutation, so the strains that affect us later will most likely be different from the ones to which we've been vaccinated. So that's why the flu vaccine is so different from year to year. Here's all the viruses from year to year that we've seen. The AIDS epidemic hit in west coast of the United States, New York region in 1981. This is what it looks like under the scope. H1N1, which is the flu that is the common flu. This one killed thousands, hundreds of thousands in 1918. West Nile virus hit, in this case, sort of Europe region. This guy was in Sub-Saharan Africa, not Sub-Saharan Africa, Middle East to us. That's why it's the West Nile virus. Severe acute respiratory syndrome in 2002. Avian flu or bird flu in 1997. Target in China. Do you guys remember that? Where you started killing all the birds and chickens. Ebola virus, this is what this guy looked like in 1976. We still start to see several variants of Ebola. Zika virus, Zika fever in 2015, and H1N1 in 2009. When you guys get finished with your exam, you are more than welcome to write an extra credit paper on any one of these flu viruses. In fact, you may do more than one to do 2 papers on this, and you are welcome to write as much as you need on that. Okay, so what's HIV? HIV is a retrovirus. A retro means going back, right? So it uses RNA instead of DNA. It uses RNA as a template to make DNA, which then inserts itself into the chromosome. So then it's not RNA transcribed to DNA transcribed to RNA, it's RNA, which is used as a template to transcribe to DNA. So totally different. That's why it's called retrovirus. These viruses carry molecules of an enzyme called reverse transcriptase, which catalyzes and speeds up reverse transcription. So the synthesis of DNA on an RNA template. So checkpoint. Why is HIV reverse transcriptase a good target for anti-AIDS drug therapy? Because it's unique to HIV. We don't normally copy information from RNA to DNA, right? What do we do it? From DNA to RNA. So disabling reverse transcriptase would not adversely affect a human. So this is what the HIV structure looks like. This is the glycoprotein plus envelope. This is the protein coat on the inside, with RNA in the inside, 2 identical strands of RNA, and then it's got reverse transcriptase in the inside. So what happens is the viral DNA has reverse transcriptase. This guy, the RNA is transcribed into DNA. The DNA is doubled. Eventually, in this case, it will go inside the nucleus. The chromosomal DNA and the provirus DNA eventually will turn into a single strand of messenger RNA, which then turns into viral DNA and RNA and proteins, which then break out into it and create more virus structure.
Understanding the structure and life cycle of HIV, the AIDS virus, is critically important to dealing with Understanding the structure and life cycle of HIV, the AIDS virus, is critically important to dealing with the disease. HIV is covered by an envelope derived from a host cell membrane. Glycoproteins studying the envelope recognize and bind to receptor molecules on the host cell. A protein coat surrounds the viral genetic material, which consists of two molecules of single-stranded RNA. The enzyme reverse transcriptase enables the virus to make DNA from an RNA template, a trick for which this group of viruses is named retroviruses. The RNA molecules of HIV enter a host cell when the virus fuses with the plasma membrane and the coat proteins are removed by enzymes. Reverse transcriptase catalyzes the synthesis of a DNA strand complementary to the viral RNA strand, and then a second DNA strand complementary to the first. The double-stranded DNA is incorporated as a provirus into the host cell's chromosomal DNA, where it may lie dormant for years. Occasionally, the provirus is transcribed into RNA. This RNA serves as both messenger RNA for the formation of HIV proteins and as genetic material for the next generation of viruses. Protein coats form around viral RNA and reverse transcriptase molecules. Viruses bud from the host cell, acquiring envelopes as they leave.
Okay, awesome. These videos are great. I would just watch all the videos in a row to study for the exam. All right, so we're going to now take a look at infectious proteins. These proteins are called prions. Prions can cause brain disease in animals. So do you guys remember mad cow disease? Mad cow disease caused basically the cows to moo. That's why they were like thinking they were going hysterical. There's something wrong with their brains. What happened was prions were actually infecting the mad cows themselves. So when the prion gets into a cell containing the normal form of the protein, the prion somehow converts normal protein molecules to misfolded versions of the protein. So again, what makes a prion different from all known infectious agents? These guys are proteins and they really don't have any DNA. Okay, this is a computer model of a prion. When you take a look at prions, what we're eventually going to study is types of bacteria like mad cow disease. You guys are more than welcome to write an extra credit paper on mad cow disease. I remember a lot of people had pictures of large 200 or 300 cows that they would burn and incinerate because they were afraid that if that would eventually be meat then that was sent to the United States. We know it started in the United Kingdom. That meat will then unfortunately infect us and obviously transfer the prions to us because we ate the meat that was infected by mad cow disease. So there's a couple things that bacteria can do to transfer DNA. It can transform, uptake the DNA from the environment. It can transduce via gene transfer by phages, bacteriophages, or it can conjugate, which is transfer from a DNA from a donor to recipient. So what happens is transformation, transduction, and conjugation is basically when a fragment of DNA from another cell enters the bacterial DNA and transforms it. A phage is when it transduces its own information and fragments the formal to create the host. Conjugation is when the donor basically starts to attach with a bridge and conjugate. This is kind of like through a sex pilus. And the sex pilus will basically start to send its donor to the recipient and infect bacteria that way as well. So the recipient cell will have the donated DNA. It'll cross over and degrade the original DNA and recombine it, which is really scary in the end. So bacterial plasmids can serve as a carrier for gene transfer. So when when the donor E. coli muscle can carry out conjugation, it's usually due to a specific piece of DNA called the fertility factor. The fertility factor can exist as a small circular DNA called a plasmid, and it can exist as an R plasmid, which poses super bad problems for human medicine because it carries genes for enzymes that destroy antibiotics. So plasmids are used for tools for genetic engineering. Why? Because plasmids can basically essentially take viral infections, and the best defense against them would be changing the virus through the use of vaccines, right? So that's why they're very useful tools for genetic engineering. And we'll talk about antibiotic resistance bacteria as well. But again, F factor or fertility factor is when something is integrated to the original cell. That factor starts replicating and transferring it into the chromosome. The recipient cell will essentially take that chromosome and then recombine later on. So in that essentially is going to create a new donor, which is that new cell. So here's the plasmids sitting inside E. coli, and this, if you blow it up, it looks like a part of bacterial chromosome that is released from a ruptured E. coli cell. So here's what we eventually need to do: 1, 2, 3 to 10 to 15 to 20. Make sure you guys are able to do this, and then use this to help you understand what happens with transcription and translation. All right, guys, I'll see you on Wednesday. Good luck with the studying. Go through those videos. Expect 50 questions with 20 extra credit covering chapters 8, 9, and 10. All right, have a great rest of your Monday, and then I'll see you folks later.